Okay, so the article I decided to do my presentation on is the development of the human Mullerian duct in the sexually undifferentiated stage. This article was written by Ryozo Hashimoto and was published in the Anatomical Record in 2003. And my name is Chanel. I will be presenting this today. So, the first thing I wanted to call your guys' attention to is that the author of this paper references the ducts as the Mullerian and the Wolffian ducts. These are kind of tombstone names, and we've started to switch over to the mesonephric and paramesonephric duct names, as Dr. Myers has discussed in class. But since the author of this paper used these terms, these will be the terms that I will use throughout the remainder of this presentation. During the undifferentiated or indifferent stage of sexual development, two pairs of genital ducts form in both sexes, as you can see in the top picture with this purple gonad, the red Mullerian duct, and the blue Wolffian duct. You can also notice in this picture that the Wolffian duct is in direct contact with this purple gonad through this kind of yellowy orangey mesonephros. And the red Mullerian duct is not in direct contact with the gonad. And we have discussed that in class. I just wanted to repoint that out. Around 6 weeks in utero, if the fetus has a Y chromosome, it will express the SRY gene, again as we have talked about, which will signal the release of testosterone and masculinize the Wolffian duct, and the gonad will become a testis. Furthermore, the SRY gene is associated with the release of another hormone called anti-Mullerian hormone, or AMH. We have also described it as the Mullerian inhibitory hormone or substance, and this is going to cause the degeneration of the Mullerian duct, which we can see in this part of the figure right here, where we have a developing male. Without this Y chromosome and the SRY gene, testosterone and anti-Mullerian hormone will not be released, and the Wolffian duct will go away, and we will get a feminized gonad, the ovary, and a Mullerian duct, which again is not in direct contact with that gonad, and this is for the developing female. The Mullerian duct is said to develop autonomously as an invagination of the coelomic epithelium, and then passes caudally and fuses into a single canal or uterovaginal primordium. These drawings remind me of ones that we have been drawing in class all semester. So in blue, I have highlighted this coelomic epithelium, and then you can see in red the Mullerian duct that is invaginating from that coelomic epithelium, and in this picture right here, we can see that that invagination is passing caudally. Okay, this study was done at the Congenital Anomaly Research Center at Kyoto University Graduate School of Medicine. They obtained 7 embryos from apparently normal pregnancies that were terminated via a dilation and curettage, or D&C, procedure between stages 18 and 23. The embryos were then stored in formalin until serial sections could be taken for histological analysis. Another thing I wanted to mention is that this article didn't give any time periods, such as days or weeks, for these stages. So all you guys need to worry about is that stage 18 is the earliest stage and stage 23 is the later stage. This is a sagittal section of a stage 18 male embryo showing the cephalic end of the mesonephros at 5 levels, A through E, and the scale bar here represents 200 microns, and F is just a more magnified version of E, where the scale bar represents 100 microns. I've also included a list of abbreviations to the left and the directional terms dorsal and caudal for some directions. You can see that as we progress from A to B, the pleuroperitoneal canal is closing and becoming completely separate from that coelom. From C to E, we can see both the Mullerian and the Wolffian ducts forming. And if you look closely at D, you can see that the Mullerian duct is actually in contact with that coelomic epithelium, again providing evidence of the coelomic epithelium invagination. In F, this more zoomed in version of E, we can see that the two ducts are so close in this region that they're actually sharing a basement membrane at the caudal end. This is a transverse section of a stage 19 female embryo at the caudal end of the Mullerian duct, where the scale bar represents 100 microns. Again, we have directional terms and a list of abbreviations provided. In this figure, we can see that the Mullerian duct is within the basement membrane of the Wolffian duct. So this M represents this Mullerian duct right here, and W represents this Wolffian duct. And you can see that the basement membrane right here is shared between those. It's kind of hard to see, but you can kind of see the different colors of the light gray versus the dark gray. This is a frontal section of a stage 22 male embryo showing the caudal portion of the two elongating Mullerian ducts at 5 levels from A to E. And the scale bar here is representing 200 microns. In A, you can see that the two Mullerian ducts, labeled as M, are in direct contact with one another. And in B, we can see that there is a septum that is separating the two Mullerian ducts, or at least the lumen of the two ducts. And from C to D to E, we can see that the separation of the Mullerian ducts from one another and the joining of them to the Wolffian duct. So they are separating here and here. And in E, you can see that there isn't really what looks like any Mullerian ducts. We just see those Wolffian ducts because the Mullerian ducts have joined with them. To the left, we have a sagittal section of a stage 22 female embryo. And to the right, we have a frontal section of the same stage 22 female embryo. In the sagittal section, you can see that the Mullerian duct has lost its lumen and basement membrane and has continued as a cell mass within the basement membrane of the Wolffian duct. For the frontal section, I had a hard time distinguishing the two ducts based off of where these letters were, so I decided to color code them to make things easier to explain. So these blue ducts on either side are the Wolffian ducts, and the two shades of pink, this darker pink and lighter pink, represent the two Mullerian ducts. And you can see here that they are separated and then they fuse together and then they separate out again. This is a frontal section of a stage 23 male embryo showing the caudal portion of the two elongating Mullerian ducts at 8 levels, where this scale bar represents 200 microns. We can see that as we progress from A to H, the two Mullerian and Wolffian ducts are migrating towards one another, So you can see there's a space in between here. That space closes in B, and then the Mullerian ducts are going to get closer together until there's just a septum separating their lumen, at which point it will look like we have one fused Mullerian duct, and we continue to fuse until the Mullerian ducts have become engulfed or completely engulfed by these Wolffian ducts, and we see that here. Okay, a study done by Grunwald in 1937 with chick embryos showed that the Mullerian duct did not grow caudal to the experimental interruption of the Wolffian duct cells, and the caudal end cells could not be distinguished between the two ducts. So this led Grunwald to assume that the Mullerian duct cells are derived from the Wolffian duct, and in all the embryos in this study, the Mullerian duct was found to be in close contact with the Wolffian duct, specifically in those cells. So this also supported Grunwald's claim. Although the origin of the Mullerian duct is unknown, both of these studies acknowledge that there is active communication between the Mullerian and Wolffian cells, and they may have a common origin, but that has yet to be decided. And with that being said, I'm happy to answer any questions that anybody has or any comments or concerns. Thank you.